Hello everyone, and welcome to today's live broadcast, Brain Tumor Exosomes and Cancer Immunity, Fat Balls of Immune Confusion, presented by Michael Graner. He is an Associate Professor of Neurosurgery at the University of Colorado, Denver. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Michael Graner. I will now turn the presentation over to him.
Okay, good day everyone. I'm Michael Graner, as you've just been told. I'm at the University of Colorado, Denver, the Anschutz Medical Campus, and although I'm in neurosurgery, I'm not a neurosurgeon, I just kind of do the science behind it. I've been asked to discuss cancer immunity relative to things called exosomes. These are little fat balls that get spit outside of cells in one way or another. So we'll move along and talk about that. First, I need to declare my disclosures. I have nothing to talk about. You will see a number of cats and dogs throughout this performance. How did I get into this is a frequent question, and it includes basically my real science career began when I left the world of Drosophila and moved into Pediatric Oncology, which is a perfectly logical progression. There I worked in a laboratory where we were developing cancer vaccines, and these were heat shock protein-based cancer vaccines. So those heat shock proteins are stress proteins, so you would have thought that we would have studied stress responses ahead of time, but we didn't. Instead, we went right to the stress proteins themselves. However, as I moved on in my life, I started studying the actual stress responses, and that is where we came across the idea and the presence of things called extracellular vesicles or exosomes. In theory, we were the first people to describe these in brain tumors, and it came from basically stressing cells. Well, that led back to the idea that we could look into the immune responses to them because we were kind of a vaccine lab. And later on we learned that those particular stress cells could also turn into, spit out exosomes or extracellular vesicles that could be immune suppressive. So the story has been kind of swinging in multiple directions, so going from vaccines to immunotherapy to cancer immune suppression. That'll all be sort of discussed in the context of today's talk. So the problem that I work on is the brain tumor. The worst ones are called glioblastomas, and they are they're pretty nasty boogers. The median survival for patients is only 15 months, and the quality of life can be pretty poor. Everything you see here is the problem with a brain tumor. There's a very heterogeneous batch of cells in there that's obviously quite proliferative. It's invasive. It's across the corpus callosum, which is the middle of the brain slice you see there. That's kind of a no-go for surgery. And you'll also see that there are intense areas of bloodiness, so they're very vascular. They're also very hypoxic. And obviously, and since we're looking at this slide, things went badly for this patient. They're actually very resistant to therapy. There are a number of reasons for that. One is molecular, and some of it is just plain physical. The idea that this is in the brain, and the blood-brain barrier is a major impediment to getting drugs up there. I should point out that surgically and chemoradiation are pretty much only standard of care therapies right now, so we're focusing a lot on experimental therapies to try to improve this completely abysmal outcome. Now, what I'm going to talk about today may be somewhat particular to brain tumors, but in general it's going to apply to most other cancers as well. So moving on, those things, those extracellular vesicles, those fat balls, this is a gorgeous slide that I've of course drawn myself. that kind of covers everything that I hope we can get out of the story. I'm going to be looking at essentially two portions of this. One is going to be what we call, change my thing here, the exosomes, and those are going to come from the endocytosis of cell surface molecules and the entrapment of cytosolic proteins within those endosomes. So that's what you're following here is as proteins on the surface are engulfed into the cell by an endocytic mechanism, and pick whatever mechanism you want, they will end up in these early endosomes. Those early endosomes then mature to later endosomes, but what happens during that period is you get this blebbing, this internal blebbing that occurs that picks up pieces of cytosol. So proteins and RNAs that are in the cytosol will now get taken in to these vesicles, and these bud off completely to the inside, where you have small little vesicles inside a larger vesicle. At this stage, the thing is called a multivesicular body, and the little things inside it are called interluminal vesicles or intravesicular bodies as well. Notice we can't have one name for this stuff. We have to have multiple names for them. The canonical fate of an MVB is to go to the lysosome and be degraded and have the contents sort of recycled. But we know that under some conditions, these vesicles will actually end up fusing with the plasma membrane. And that's, get another thing to draw here, right here, for instance. So what that does is it releases the little vesicles from the inside to the outside of the cell. And now we call those exosomes, and that is the official designation for them. Other means of having this occur is when things will split off. Try to get a change of this. We'll split off the cell surface directly. They'll bud off like that, and they're called microvesicles or ectosomes, or sometimes shed vesicles. Notice we can't have a single name for anything. We have to have multiple names, and nobody agrees on these things. So the point that I want to make from this is that in general these are they look like the cell itself. They've got a rough recapitulation of the outer membrane and the inner membrane. Proteins that were sticking out of the cell membrane are going to be sticking out of the vesicle membrane, and proteins and RNAs that are inside the cell will end up inside the vesicle. So there's sort of a recapitulation of what the cell looks like on a very tiny scale. So we think of them as means of intercellular communication, since they can leave the cell, and instead of having cell-cell contact, you can now have cell-vesicle contact. So the vesicle leaves the cell and goes and bumps into another cell and transfers information to it. Now these were originally discovered by Phil Stahl and Rose Johnstone. They're the ones credited for giving them the names exosomes. And it was as a form of disposal, the idea that as reticulocytes were maturing to red blood cells, they had to get rid of things like the transferrin receptor after losing their lysosomes. So that was the idea was these are essentially garbage bags. That's not a very sexy concept, except when you're living in a place where they don't pick up the garbage, and you understand how important that is. We have studied a lot of the effects of signaling, as have other groups, the idea being that the vesicles can carry signaling molecules themselves that can be delivered to recipient cells, but they can also prompt changes in signaling on those recipient cells. And one of the big areas that's come out lately is the idea of RNAs that are inside these exosomes or extracellular vesicles being transferred almost in a viral-like fashion to recipient cells. And speaking of viruses, they have figured out this pathway long before we did. So viruses really understand how to hijack this system and take off, as do a number of other pathogens. They're way ahead of us in terms of how we study these. Oops, trying to make this go forward. And so there are my drawings, then there's other people's drawings. So this is these are the much improved artwork over time. So we see, for instance, here, extracellular vesicles moving towards inside the multivesicular bodies, being released outside, essentially a farting mechanism. Over here it looks like a zit being popped as these things are squeezed out. And here we see the bag of worms version of this. So these are extremely crowded, very heavily laden little balls, or tiny little fat balls. And as we have noticed, not all of us got the message about the artwork. I promise it's the last time you'll see that slide. So one other form of extracellular vesicle that I didn't mention, but we don't talk too much about them, are the apoptotic bodies. So I've described the exosomes coming through the endosomal system, the microvesicles that pop off the cell surface. And we've included in the term extracellular vesicles the idea of apoptotic bodies. So as the cell sort of degenerates, it will release these things. They're usually considered to be pieces of material that are taken up by the neighboring cells, but it turns out they have some very interesting capacities as far as also signaling and talking to those cells, but we're not going to discuss those. Other critters make these things as well. So single-celled organisms will make exosomes and plasma membrane blebs that are essentially the same thing as microvesicles. And gram-negative bacteria make outer membrane vesicles. So I've got to point those out. I also need to point out this particular review article because I'm one of 60 authors on it. Very proud of that. The idea, though, I want you to make sure you get is that cells don't release simply one kind of vesicle. So we need to bear in mind that there are going to be multiple spheres of multiple sizes and multiple origins floating out of a given cell at any time. And as far as we know, all cells make these things. Now, to give you some idea of how big they are or how small they are, here's a nice slide from a review article, and it puts them in the context of things you probably recognize or may have heard of, such as bacteria, platelets, protein aggregates, and the exosomes, which are the ones I'll mostly be talking about, are on the size of viruses. And so this is the appreciation of dimension. It's not a terribly accurate appreciation of dimension. It's more like this. So when we're starting to talk about the different sizes of vesicles that can come out of the cells, this is relatively what they tend to look like. So I'll be speaking mostly of the exosomes down here, which are, like I said, on the size of viruses. As far as those dimensions go, the literature is sort of replete with different terms that people have used, mostly depending on who's reviewing your article. So we've commonly said they're 30 to 100 nanometers in diameter, once again, sizes of viruses. I've seen in the literature these numbers changing and going up over time, and I think it's because people don't really know what they're talking about. So when you get to the size of about 300 nanometers, you're starting to talk about something that's going to actually fill the multivesicular body, so it's probably not a true exosome. So if we focus on these 30 to 100 nanometers as sort of the classical size, what does that mean in terms of a volume? I had a student sort of delve into this, and he came up with the term yoctoliters, and he didn't invent that term. It's real. I'd never heard of such a thing, but it's about 10 to the minus 24th liters, so it's pretty tiny. But once again, the concept that they're about the size of viruses suggests that they can hold about, packed tightly, 100 proteins and sort of 10,000 nucleotides worth of stuff, of nucleic acid, just to give you some idea of how much stuff is in these things and what we do about that. So in other words, they're packed pretty tightly, and we now know from some of the really good cryo-electron microscopy work that's going out there that these don't necessarily have to be round. We always draw them as round, but they don't necessarily have to be. And also things will flop out over the sides, and those are very important because those are probably ways that they talk to other cells in terms of either bouncing off them or being taken up by them. So what's inside and on the outside of the exosomes really matters. So as far as the contents of an exosome go, this is sort of a standard view of it, and I'm just going to highlight some of the things that we almost always see in them. From proteomic analysis, we almost always see metabolic enzymes. We almost always see annexins. We almost always see heat shock proteins, very typical stuff. Also, the tetraspanins are sort of our method for identifying these. Pick a CD something, CD9, CD81, CD63, all of these are regarded as tetraspanin-type markers for these extracellular vesicles. We also always find presumably within the vesicles, actin and tubulin that we assume is monomeric because there's not a lot of room for polymerized particles to be in there. So that's probably one of the issues we would ask is that most of the proteins that get in seem to have been in the form of complexes, but oftentimes those complexes are too big to actually get in the cell. We also see the RAB-GTPases. Those are sort of classically another way to define these. Now, depending on where the exosome came from, what cell type released it, we may see different types of receptors. We may see different types of adhesion molecules. You may see TGF-beta. We also may see major histocompatibility complex molecules because those are produced by most every cell. Serum proteins are often regarded as contaminants because they cling to these things, but biology doesn't really like the idea of a contaminant, so these are probably real and probably belong there. The types of lipids we see in them are usually structure forming because these things have to bend very tightly, and it takes a fair amount of energy to do that. So you're going to have structure forming lipids as well as bioactive lipids. So that's a rapid overview of the protein content. Now the RNA content I will get into a little bit later. Any kind of RNA you can think of pretty much shows up in these things, which means there turns out to be RNA binding proteins, ribonuclear proteins, ribonuclear particles that associate with them as well. And both single-stranded and double-stranded DNA have been found in them also. So which, when you think about that, if you're able to isolate these vesicles, you might be able to determine the, say, mutated state of a person or of their cancer, so to speak, based on the DNA you may find in them or even the RNA you find in them. I'll talk a little bit more about that later. So when we got into this, we'd done a lot of proteomic work, and we were noticing, as I mentioned, the number of nucleic acid binding proteins. And that prompted us to think that we needed a larger cat's eye view of the situation to see, well, what sort of proteins have been identified in these. And fortunately for us, there is a database called Vesiclepedia that houses pretty much everything we know about the protein contents of these vesicles. And when we sifted through that to get rid of all the duplicate entries and just identify proteins we could come up with that were human, the reason we used human is so we could use them in downstream sort of analyses that are more prone to human studies than anything else. We ourselves came up with a little over 3,000 proteins, and this is just a table showing you some of the work we did with that. A much better job was done by these guys, by Young Song's group in Korea. They identified around 4,000 proteins, the 13,000 vesicle proteins total, but of those, about 7,000 are found in humans, and of those, about 4,000 are the most prevalent. So we're looking at a subset of the proteome in terms of how many genes and how many proteins could possibly be in there. It doesn't seem like everybody gets in, but there tend to be some sort of selection methods. We don't really understand what those are. One of the other really important components of these vesicles are the lipids. Obviously, I'm kind of wound up about those because it's necessary for these lipids to work to fold and bend this thing into this very tight structure. Among those, you will find the structure-forming things such as ceramides and cholesterol. The reason we don't have a great concept of what the lipids are is because those technologies for identifying them are far behind what we have for, say, the nucleic acids, the proteins, so that just the databases aren't there, the techniques aren't there, the libraries aren't there, but they're growing. One thing to imagine about that is you'll have a particular head group of a lipid that can have multiple or different chain lengths, tail lengths on it, and those can be multiply saturated. So you can imagine trying to do mass spectrometry on that becomes pretty difficult when you've got so many variables in potentially the same molecule. So I'm not going to talk too much more about that, but I will say that these lipids are somewhat specific for the vesicles. There are distinctions between what gets into the vesicles and what is coming out of the cell. Like I sort of acted like it's the exact same thing, but there is a selection for that. One thing we've delved into, and this is slowly becoming more and more popular, is the use of metabolomics on these, which enables you to identify not just, say, lipids, but also sorts of aqueous molecules as well. And this is just some work we've done. The one thing that I found interesting about that is that we can distinguish between the metabolomes, so to speak, of cells versus the exosomes using principal component analysis. So there is something distinct about what gets in these as cargo, not only as proteins and lipids, but also as other types of metabolic phenomenon. So we're still pursuing that, and lots of other people are too. Now to the RNA world. About 2007, Jan Lutval's group identified RNAs in extracellular vesicles in these exosomes, and the RNA world went nuts. People loved it. It's a viral component, and even the NIH Common Fund threw money at it. As someone once said, it was as close as the NIH could come to a genome, and so that's where a lot of money went. That's where a lot of interest has gone. You can see over here that every conceivable and some not so conceivable RNAs, including miscellaneous RNAs, show up in these things, have been identified in these vesicles. And my take on that is if you identify an RNA, throw a letter, 2 letters, 3 letters in front of it, you've got a publication. There's also some DNA in there. But as far as the RNAs go, the one thing I would really like to do is identify something and call it RNA, and thus cause an existential crisis. They will love that, trust me. So where do these things come from and where do they end up? Well, cells make them, but they end up in biofluids. And this is how they get transported from one place to another. This is where we will find them in the media from cell lines. We'll find them in blood. We'll find them in urine. We'll find them in saliva. Other places would include synovial fluid, bronchoalveolar fluid, cerebrospinal fluid, semen, breast milk, things like that, and especially in the tears of scientists after they get their grants rejected, very loaded with exosomes. One thing that I should point out is the cryo-electron microscopy area has become very big, obviously people winning Nobel Prizes for this, compared to what we see with these usually flat squish things that look like red blood cells that have been run over by a truck. The amount of detail and information in the cryo-electron microscopy is just extraordinary, and I think this is going to be a huge benefit for us to start understanding the external structures of these as opposed to just seeing them as flat circles. So if these things leave the cells and end up in the biofluids, where do they go next? Well, they can go into your liver, your spleen, your kidneys. You can urinate them away, you can poop them away, they can get degraded in various places, but they also run into other cells, either nearby or far away. As I kind of mentioned, any mechanism that you can think of to have these internalized, whether it's directly fusing with the membrane and just allowing the contents to enter inside, or any form of endocytosis, whether it involves clathrin or not, caveolin or not, receptor-mediated endocytosis, so this is another one. Any way you can think of to get something inside a cell is a way that a vesicle can get inside a cell. What happens after that is not so clear. They may be degraded, they may be stimulatory, they may be able to promote things such as the delivery of these RNAs. So what happens after that is a little unclear, except we watch for the responses of the cells, and that's sort of how we spend most of our time from now on, is throwing exosomes on cells and asking what do those cells do. So we view them as information packets, ways to talk. Cells can talk to each other nearby, far away, and have influences that are probably seemingly greater than you would have if you just had cell-cell contact, especially if those cells can't be in contact. As I mentioned at the start, I was kind of asked to put this in the context of the immune system, and I, in a paper I put out some time ago, was asked to do the same thing and say, show me the immune system in one slide. So that's what this was about. The immune system at the most basic level consists essentially of things that we would call innate responder cells. Excuse me. These are cells that are primed to go after something when something goes wrong. So you get a bacterial infection, they're there. A viral infection, they're there. On the other end of this, the adaptive side of things gets a little more specific. It says I need to find a particular antigen basically to go after, and that can include things in antibodies and in T cells. The link between them are antigen presenting cells. These are things that recognize problems in the sort of an inflammation activity and then telling the adaptive immune system something has gone wrong, we need to fix that. So the cells we'll be focusing on in this case will be B cells, T cells, and NK cells for pretty much the rest of the talk in that sense. The concept of immune surveillance goes back to the 1950s, actually. So McFarland Burnett and Lewis Thomas were the big purveyors of this, and our understanding of how this works has changed over time. But the basic concept is the same. The idea that neoplasia is perhaps common, but cancer itself, relatively speaking, is rare. So there must be some sort of immune mechanisms that look for things going wrong and then try to stop it before it gets out of hand, and that's considered to be the form of immune surveillance. So we presume this works and keeps cancer out of our bodies most of the time, but obviously things change, and that's where the immune failure comes in. We know that tumors can actually literally fight the immune system by throwing out particular cytokines, putting up barriers, essentially attacking the immune system. So there is an outright battle going on in that case. tumors also can hide from the immune system. They can cloak themselves in proteins that look normal. They can cloak themselves in other normal cells. And there are particular molecules that the immune cells look for to recognize what's going on inside another cell. Tumors will actually downgrade those and try to get rid of them, so they appear basically invisible to the immune system. And then eventually the immune system begins to ignore the tumors and perhaps suppresses itself, which only allows the tumors obviously to grow, essentially unstopped by the immune response. So as I had mentioned before, and this is yet another one of those gorgeous drawings, the idea that these little vesicles look like tiny portions of the cell, little cellular surrogates. I actually wrote that in a book chapter. Does that mean we can use them? Does it mean we can use them, for instance, as biomarkers? If the cell is sick and they're releasing vesicles and we can identify essentially implications in the vesicles suggesting that the cell that spit them out is sick, that can be used as a biomarker to identify a disease. On the other hand, it can also be used as vaccines. In a tumor setting, these are little antigen-laden fat balls that get spit out of them. They're just loaded with antigens. We could be able to use them to provoke an immune response against the tumor. As it turns out, they're also loaded with immune suppressive molecules. So it becomes a question of, are they useful or are they not useful, and how can we determine this, and what is this dichotomy? So let's go to the vaccine side of things. And oftentimes, when you hear the term tumor exosome, it's called TEX, and this will pop up a little bit later. What's on TEX? TEX has all sorts of cool things. On the inside of the exosome, there are presumably antigens, antigenic materials, mutated proteins that come from the tumor that could be recognized as not self, if that's what the immune system sees. Also, heat shock proteins. As I kind of mentioned before, my vaccine career got started with these things. We understand that they carry little pieces, little peptides of the tumor proteome, and if those are mutated, those are nicely presented by the immune system to from the antigen-presenting cells to the T cells to start driving immune responses. On the surface of these things, these heat shock proteins and also things like the high mobility group proteins are seen as danger signals if they're released outside the cell. And the tumor can also literally put out things called the major histocompatibility complex molecules that have peptides bound to them. So if that's what's going on, and this runs into a tumor, or I'm sorry, it runs into a dendritic cell, there are several things that can happen. That internalization process that I talked about before, that occurs. It can release directly to the inside. And this forms what is kind of the sort of black box of antigen processing. So these proteins are now released into the dendritic cell, and it can take them up, chop them into pieces, and end up presenting the little peptides of those on what we call major histocompatibility complex molecules 1 and 2. These talk to different cell types. The MHC1 talks to CD8 T cells. These are the killer T cells. MHC2 talks to the CD4 positive T cells, and these are called the helper T cells. Now, the stimulatory effects of these, say from the heat shock proteins or the HMGB1, excuse me again, will actually prompt the T cell, or the dendritic cell rather, to make co-stimulatory molecules. So these are things that poke and prod the T cell. Once that T cell of destiny with a T cell receptor recognizes a peptide antigen, it can now get stimulated by the dendritic cell to go run out into the periphery, reproduce, and go seek out offending cells that are displaying the wrong molecules, basically, antigenic things out on their surfaces. And as I kind of mentioned, most of the time we expect that the vesicles get internalized and released, but sometimes the vesicles can just park on the cell surface, the dendritic cell surface, and essentially act as extensions of the membrane, triggering these immune responses. So that's the goal of the concept here, is that these vesicles contain antigen, but they also contain stimulation, and that prompts these antigen-presenting cells to get wound up, present antigen to T cells, and get them wound up, and have them go out into the periphery and chase down the things that are wrong, send in the cavalry, cure cancer, and that's what we're after there. I should also point out that they affect the natural killer cells, the NK cells, in multiple ways. So that they often will display, these vesicles will display things like MICA1, MICA-A, and MICA-B, and dendritic cell exosomes, which we call DEX, notice how clever we are, TEX and DEX, put out things called ULBP1, binding protein 1, as well as IL-15 receptor alpha, which literally hands off IL-15 to the IL-15 receptor beta and gamma. And then the heat shock proteins that are outside can actually stimulate a surface receptor called CD94. All of these things get the natural killer cell wound up. It releases granzymes. So these are basically bags of proteases that are going to go and punch holes in the neighboring cell. They spit out pro-inflammatory cytokines, TNF-alpha, interferon gamma. They upregulate receptors for antibodies. So if the cell next to this has an antibody stuck to it, because you're generating an immune response, the FC receptor will latch onto that and stimulate antibody-dependent cellular cytotoxicity, which I can't see because I'm making a mess of my slide here. But in any way, so that will then start punching out the neighboring cell, trying to kill it off as well. So that's what the natural killer cells will do. Now we showed in this particular paper that T cell and B cell responses were pretty potent, and as a vaccine, when you use these things, so we had mouse models here, can't draw. So this particular brain tumor mouse model, and if we vaccinated mice with that, we didn't vaccinate mice, tumors grew. If we did vaccinate mice, tumors didn't grow. Well, there's one in every crowd, that one. If those mice don't grow tumors, they tend to survive. So we saw enhanced survival for these things over time as well. Now this is done in a prophylactic setting where we were able to give the vaccine ahead of time. As it turns out that if you don't do that, if you do it in what's called a therapeutic setting, implant the tumor first and then try to treat, and this is a very stringent model, it was difficult, we didn't see these responses. So we had great responses in the context of no tumor until you tried to give it a tumor, so protectively, but it wasn't useful in a therapeutic setting. So this was kind of disturbing. What we realized, first of all, that the T cells and the B cells are very important. We didn't study the NK cells. But the idea then was, well, we've got this great idea. We'll stimulate the stimulators. We'll affect the effectors. We will write grants. That didn't go so well. The National Cancer Institute basically told us that, no, it's all about immune suppression. And despite all of these data and all of these papers that you see here, you're just wrong. So the pendulum had swung the other way. We were now seeing these tumor vesicles as immune suppressive. And that meant we kind of had to change gears, but also we still would bear in mind that they were good vaccines. What's the difference between the vaccine potential, the stimulatory potential, and the suppressive potential? So what are these suppressive effects? Well, not only do the tumors make the good things we want to see, the antigens, the HSPs, stuff like that, they also put out things like the RNAs. We don't know a lot about what these do, but presumably some of them will be suppressive to receptor cells like T cells. As far as there are these ectonucleases, CD39, CD73. T cells love ATP. These things eat ATP and convert it to adenosine. T cells hate adenosine. So that's a rough one. There's a molecule called Fas ligand that's present on a lot of tumors and ends up therefore present on their exosomes. It's known to interact with the Fas receptor. This ends up killing these cells, literally killing those T cells. It's a death receptor, and it's not a pretty sight. So we were going to move on from that. Other things that you may have heard of over time because they're getting more and more famous are things like the program death ligand 1, which can be on the tumor and therefore is on exosomes. It interacts with program death 1 ligand, or receptor rather, on the T cells and essentially disables them. Transforming growth factor beta is also present on the surface of these things in a variety of forms, sometimes completely latent, sometimes completely active. That's able to take T cells and essentially turn them into regulatory T cells. So regulatory T cells are the no-fun police of the immune system. These are very potent cells in terms of shutting off activated responses as well as shutting off the initial signaling that goes through dendritic cells. They're very potent at that. On the B cell side, so these surface antigens are very good. For instance, we learned that they make some remarkable antibodies in recipient cells, but unfortunately those antigens can also titrate out those antibodies that may be either generated by the host or things we actually use as therapeutics. And obviously this reduces the ability of the immune system to use the antibodies against the tumor. Another aspect of these things is they make complement receptor, which takes complement out of the equation and therefore reduces complement-dependent cytotoxicity. Some of these danger signals, if overused, will end up making regulatory B cells. So these are kind of like the no-fun police of the B cell system. So think of them as these molecules are on the surface of the cells. They can stimulate, but they can also suppress. And I think that's where we need to understand the main differences on them. As far as the natural killer cells go, those same proteins that were very good at binding receptors and stimulating the NK cells, such as the NKGD2 ligand and the CD94 ligand, those are also the same ones that will end up causing them to go away. So they will cause the NK cell to pull those out. And that's part of the safety mechanism, right? It's part of keeping the NK cell from going crazy and killing everything it sees, because it doesn't really have the level of specificity that the T cells do. In addition, other suppressive moieties on these exosomes, such as TGF-beta, can actually then slow down and incapacitate the natural killer cell and essentially taking it out of the system. So the idea being that the exosomes may look like bits of tumor, but if they look like too much of the tumor, the T cell will shoot itself off. So they can appear to be decoys as well as capable of destimulating the natural killer cells. So we have this dichotomy, this yin-yang, this double-edged sword of exosomes as stimulators of the system, but also as repressors of the system. So what is this dichotomy? And we had come up with the idea that TEX, so cancer exosomes, TEX, in terms of context, we wrote an entire paper built around that title. The issue that you're going to see here, though, is that I would suggest is who sees them first. If the antigen-presenting cells see them, and they would in a vaccine setting where you're giving something usually intradermally or subcutaneously, it's going to encounter, the cells that it's going to encounter are called Langerhans cells, and they are sort of the dendritic cells of the skin. Since they've not been suppressed in any way, they're going to find these stimulatory, they're going to find antigens, they're going to promote T cell responses. On the other hand, if the tumor cell is secreting them and they get into the bloodstream, the cells that the exosomes will encounter are going to be the T cells and possibly monocytes as well. And these will have essentially destimulatory or suppressive effects. As the monocytes get turned into things called myeloid-derived suppressor cells, there's a suppressor built right into that. They'll secrete molecules that will deflate the immune system. And as I mentioned before, the T cells are being turned off as well. So who sees it, when, and how is probably the critical aspect of what determines whether you've got a vaccine quality or a suppressive quality. Now, we've done some studies on this where we, in brain tumor cells, so these are cells grown in what we call neurospheres. This doesn't really matter to most of you, but it's really cool for the brain tumor people. We take the exosomes from that and threw them on lymphocytes and said what happens to the lymphocytes. What we noticed was that there was dysregulated signaling that occurred, and this was kind of disturbing in what it led to, because it suggested that the cells would be inactivated but would still proliferate. And that's kind of counterintuitive to what most people think of in immune responses for T cells in particular. Usually their proliferation is indicative of their stimulation. But we saw they lost their pro-inflammatory cytokines and made anti-inflammatory cytokines. And then they also stopped moving. They just didn't crawl the way they should. From an immune perspective, from an immune suppressive perspective, that's kind of a novel form of immune suppression. If the T cells can't get to the tumor or can't enter it, they're not going to cause any damage. So we saw this, we published this a while back, and I've referred to it as turning T cells into teenagers, because they're living in your basement, they're eating you out of house and home, and they're not doing a damn thing. So if you've got kids, you probably understand that better than most. So that's my concept of what goes on. It's not necessarily wiping out the T cells, but just making them ineffective and metabolically kind of useless, because they're going to mess with you. So following up on these questions, one thing I've mentioned before, it didn't work, that's not showing up the way I thought it would. So we've also done some work where we've looked at what happens. I've got a thing pulled up on my screen that won't go away. There we go. We have not, I'm not going to talk about this, but when tumor cells throw exosomes back on themselves, they get wound up and make all sorts of strange signaling arrangements that they just shouldn't be doing. So, but I'm not going to talk about that. We don't have time. But I will talk about the idea that tumor vesicles will tell astrocytes, brain tumor vesicles will tell astrocytes that they want to change and become gangsta wannabes. And I'm sure all of you are familiar with the cult film Pup Fiction. That's Pup Fiction, by the way. So what are astrocytes? I am not a neuroscientist by any stretch of the imagination, which is amazing to work in a brain tumor field. They are sort of the glue of the brain. These are structurally very important. They're very supportive, not just structurally, but also metabolically and with paracrine secretions for the neurons and for the vasculature. They kind of hold the brain together. So when people say you only use 10% of your brain, what they're really saying is that a lot of it is actually structural support. From the brain tumor world, this is about as close as we get to a true glioma microenvironment. We don't have the cancer-associated fibroblasts. We don't have the cancer-associated adipocytes that would go along with this. Whoops, what did I just do to my screen? Okay, there we go. Sorry about that. Hit the wrong button. All right, and so consequently, I'm going to call them the glioma microenvironment for whatever that's worth. These are some lovely slides from people who actually know how to make really good pictures, showing, for instance, the interaction of astrocytes with the vasculature, the interaction of astrocytes, these stellate stringy cells with the neurons. And here's a nice drawing of that, these feet that are necessary to hold the blood vessels together to keep the axon stable. So that's what the astrocytes tend to do at a physical level. So we did the same sort of experiment I just described, where we take brain tumor cells and grow them in culture, and then collect their exosomes, and then throw them on astrocytes, then ask what happens. What we saw was that the astrocytes, these are normal astrocytes, spit out an extraordinary cytokine milieu. They developed these crazed signaling pathways that were very disturbing, and this led to inappropriate growth and migration. So those are going to be the main take-home messages from the last part of this talk. You're required by law, if you're in the exosome or extracellular vesicle field, to show your purification system. So here's ours. You can see it's mostly a bunch of centrifugations and filtrations. And what we get out of that are vesicles. These are cells over here, by the way. These are not the vesicles. These are the cells grown as neurospheres. What we will see, though, is that using something called nanoparticle tracking analysis, we can actually get sizes on these things. We can actually get concentrations of them as well. So these are just vesicles from these particular cells. There are a number of ways to do this. So those are just characterizations of these. We've also shown that they have all the biochemical properties in terms of molecules that they're supposed to have using these arrays as well. So there's your exosome purification. These are other methods. Most of them involve forms of centrifugation. We also have forms of electron microscopy, atomic force microscopy, all these sorts of things to characterize them, as well as the biochemical and cell biological ways to do that. The one thing I will point out is that no matter what you do, no one else is going to like it. This is kind of amazing in the field how only my way is the way to do it. We're trying to come up with a uniform method for these, but I think it's going to be difficult because the downstream, first of all, the upstream, where do we get these? Do they come from urine? Do they come from ascites fluid? You can't start the same way, and depending on what your end assay is, you can't necessarily finish the same way. So we're still engaged in how do we fight this sort of thing. So what we've done is ask questions about what happens to the astrocytes, like I said, when we throw exosomes on them from tumors, and they migrate. So if we take the astrocytes growing in a dish and we scrape a needle across it to the scratch and sniff assay, and we throw exosomes on those exosomes cause the astrocytes to fill the gap faster and way more efficiently. So it's as if the tumor is calling out to the astrocytes to come to it, come to the holes. Well, why would it do that? It's because the astrocytes then essentially feed the tumor. They put out this extraordinary cytokine array, and this is a heat map. So what you're seeing down here on this side is at 0, so it's kind of cool. What you're seeing up here at 24 hours is very, very hot. And the molecules I've outlined are things that are interesting to brain tumor people, so you don't need to get too buried in that. But just notice that everything is turned on seemingly. Interleukins, fibroblasts, growth factors, regular growth factors, all sorts of things that are just going to be yummy as far as a tumor is concerned. And we chunked all that information into some of these biopathway analyzers, in this case Ingenuity pathway analysis, and it shows us it looks like an inflammatory response. I would agree with that. It looks like cancer. Cell-cell signaling and cell migration, these things should be going on, and we suspect that's undoubtedly the case. But as far as the feeding the beast aspect of it, we took the exosomes from tumor cells, threw them on astrocytes, let the astrocytes percolate that for a while, then have them collect that media, and then we put it back on the tumor cells, that spent media, that conditioned media, and said you grow faster. And the GBM cells do grow faster. The tumor cells grow faster. And this is saying a lot because these cells grow very fast as it is. So back again to the idea that the astrocytes end up aiding and abetting the tumor when they get in contact with these tumor astrocytes, or with these tumor vesicles. And when we look at a couple of the signaling pathways that get turned on there, we see ERK and AKT are sort of at hubs of that, which prompted us to ask the question, what goes on with the signaling? So we delved into that as well with a signaling array. This is a phosphoprotein array. So what you're seeing here are phosphorylated things. So you see early on and at low concentrations, not much is happening. But as time progresses and as we throw more and more exosomes on, we see dramatic changes in a lot of these signaling molecules, including the ERKs, the STATs, the AKTs. These all seem to get turned on. And I looked at that and I thought, this looks a lot like a tumor signaling profile. And I wasn't alone in that. We chunked it into ingenuity pathway analysis. It told us the same thing. So bear in mind, these are astrocytes that now look like they have cancer is what it amounts to. They have cell death and survival mechanisms in place, and they, in theory, would look like a tumor. That was very disturbing to us, and the other thing we found is that if we take these various pathways and pile them on top of each other, we find AKT is the hub, the very center of all of that. And this is the driver of this giant hairball of stuff, which is very common in a lot of cancers, in particular in gliomas. We know that many pathways are AKT driven. What really disturbed us, though, was that if these astrocytes think they're a cancer, and we grew them in something called soft agar. This is a way to essentially define transformed cells. So if your cells grow in soft agar, they're not, and generally they don't like to grow when they're non-adherent, but we saw that actually occur at the high doses of vesicles. So it looks like the astrocytes really do think they're a tumor, at least in this vague kind of assay. Now we've done most of our assays based on arrays and things like where we chose what questions to ask. We wanted to broaden this to a little less biased form, and so we're asking about what is the transcriptome doing. So from that transcriptome, we're going to have astrocytes doing nothing. We're going to have astrocytes that were incubated with tumor exosomes, and we're going to have astrocytes that are incubated with epithelial cell exosomes, normal cell exosomes. And then we extract RNA from everything and then run it on a cool little array called the Clarion one. This one has a bunch of transcripts, so we get a lot of data out of it. Why do we say, for instance, do that as opposed to RNA-seq? This began as a student project, so we couldn't spend tons and tons of time waiting for the bioinformatics. We could kind of do this ourselves. Also, these are normal cells. They shouldn't be full of mutations, so we didn't really need to sequence that kind of thing and care about that. And we didn't really think about the non-coding RNAs, but we're beginning to do that now because it's becoming annoying. So this is preliminary data. Do not buy or sell stocks on this basis. And once again, it helps to be able to avoid your bioinformaticists when they're not terribly helpful and very demanding instead. So here's this typical sort of representation of that. Things that are falling off the line here, off the diagonal, are going to be things that we care about that are highly expressed, for instance, on the tumor side. So where the astrocytes received tumor vesicles, we're seeing what's happening up here in that corner. If we took that information and chunked it once again into ingenuity pathway analysis, we see a number of things pop up that might have been not surprising, such as interferon signaling, but the fact that they seem to think they were infected, we seem to see these inflammatory and anti-pathogen tails coming up, so we had to ask, well, is that actually going on here? So once again, these responses are kind of built into the system. You see a lot of interferon, you tend to see this the network's going to show up being inflammatory and antimicrobial. The molecules we saw, every one of these is either induced by interferon, induces interferon, or is responding to interferon one way or another. So when we look at the next step in that, just some Western blotting of some of those. We've done more, and I couldn't manage to get into this talk in time. Things called OAS, oligoadenylate synthase, such as these two. We actually have one for OAS2 as well. Goes up for 1, not so much for 3. Definitely goes up for 2, like I said, not shown. And then others glial fibrillary acidic protein. So as astrocytes, they make this, but they also tend to overexpress it if they get stressed or crazed. And we see this in brain tumors, but usually it's because of what we call the glial scar. A form of gliosis is occurring because you have a physical structure crashing through the astrocytes. In this case, it's just exosomes causing this upregulation of the GFAP protein, which is, once again, since suspecting that we've got wild stresses going on. So we saw a lot of the interferon signaling that I kind of mentioned before. We see interferon gamma, but in this case we didn't really see much. We didn't know about the type 1 interferons, alpha and beta. We knew that STAT got turned on, STAT1. We didn't know anything about STAT2. But everybody down here is an interferon responding molecule, either will further drive more interferon or just reacts with it. So looking further into that, like I said, we knew about the gamma interferon. We didn't really know about the type 1. So we did those assays as well. And what we can see here is that, look at this one's nice. It gave us both type 1, 2, and 3 in some cases, not all of them. Interferon alpha does go up. Interferon beta does go up. So these are being spit out of astrocytes when they receive brain tumor exosomes. You throw brain tumor exosomes on them, they will make they will then make these cytokines. The only one that occurs no matter what kind of exosomes you throw on it is IL-6, and we're interested in that as well. But there are forms of interferon I didn't even know existed. I didn't know that there was an interferon lambda, an interferon omega. What are these things? So all of these sort of super responses in an interferon mechanism seem to be turned on, very, very pro-inflammatory. The concept being that this looks like a viral infection. It's as if the tumor cells themselves feel like they have, or I'm sorry, the astrocytes feel like they've had a viral infection. Where would that come from? Well, as I mentioned, when we first saw this oligoadenylate synthase molecule pop up, I thought, well, that's an enzyme, nice. But it's actually a cool enzyme. It synthesizes 2' 5' oligo A, which activates RNase L. RNase L then cleaves double-stranded RNA here and here, and then these bits of double-stranded RNA are very stimulatory towards things called RIG-I and MDA5. In data I didn't show, we saw RIG-I goes way up as well. And this is part of what we would call the innate response system inside the cell. So this triggers a lot of activity at the mitochondria to start doing things, and in the end you end up making more and more interferon. So the whole interferon story comes back to this again. Where do these double-stranded RNAs come from? Probably the exosomes. And I should also point out that usually these RNAs have strange modifications to them, usually because they're being degraded. So that may be the difference between the tumor vesicles and the normal vesicles, is that the RNA is kind of battered inside the tumor vesicles, or there's just more of it. And that will lead to the this whole process of acting like you've got a viral infection, throw out interferon, try to save the day. So once again, the idea that we treat tumor, we take tumor exosomes, throw them on normal astrocytes, and ask what happens. So the cells grow inappropriately, they migrate in ways they probably shouldn't, so come to the dark side. The cytokine soup feeds the beast, and the crazed signaling suggests to the astrocytes that they might be a tumor. And that is, in terms of, say, a recurrent tumor setting, so what happens in your average glioma treatment is we cut it out, we irradiate it, and then we throw chemotherapeutics at you. So if the astrocytes left behind have been receiving, basically getting hit by tumor exosomes for a long period of time, they may be changed, and that's what we're trying to say. So exosomes in general from tumors are going to be used to benefit the tumor. That's pretty much all there is to it. Even though I tried to say that we might be able to use them in an immune setting, they're more likely, by the time we find the tumor, to be immune suppressive. And this can happen to the tumor itself, can happen to the immune cells, can bother the neighbors, the normal neighbors. And in that environment, the astrocytes are going to crawl in response to the tumor exosomes. The signaling pathways are going to change inside the astrocytes, reminiscent of the tumor itself. The astrocytes are going to spew things out, aid and abet the tumor in its growth, which is the last thing we really need. And they will turn on this amazingly inflammatory interferon response, which I'm not sure what that means. We're trying to hack into that a little bit further. And the question I would suggest or have is that these are astrocytes are a mutation away from perhaps actually becoming truly cancerous. So if we peel off that hairball down one layer, what we see, the next sort of big node down is P53. This molecule is mutated in about a third to half of high-grade gliomas. And we treat these gliomas, remember, with, we fry the resection cavity with radiation, and then we essentially bathe you in a DNA damaging agent. So I would suggest that if you've got if you drive a mutation in those astrocytes and you then pound it or have it pounded preemptively with tumor exosomes, it may be a short step to becoming an actual tumor. So what do we do about this? Well, I've kind of mentioned the AKT story. This has been a therapeutic target forever, and it's just not gone well. In many cases, because of the blood-brain barrier issue for brain tumors, it's just a hard thing to treat. Pediatric cases, they've tried a couple of things, and what they found were mostly toxicities. And there are other sort of experimental ones out on the market. So hopefully we can maybe get through going after AKT. I'm not going to hold my breath on that. But what about inflammation control? What if we started saying after we cut the tumor out, maybe we should start working on trying to keep inflammation down inside that brain area? We know that there are antibodies used against interferons and against the receptors that have been used in multiple sclerosis, and these are drugs that could be repurposed to target the STATs. But also we know that this could also perhaps be controlled by nonsteroidal anti-inflammatories, resveratrol, or curcumin. So take your nonsteroidals, drink your wine, and be sure to eat spicy foods. Maybe this will all help control that inflammation, at least at the area of the tumor. Back to the immune thing. So what's going on with that now? Well, we know that, and many of you probably know, that on the market right now is the big idea is to use checkpoint inhibitors, block those things that block the suppression. And this can come in multiple forms, things that target specific molecules, things that target the regulatory T cells. And a number of these have made it into clinical trials and have actually made it into FDA approval for a number of tumors. So it looks like the concept of suppressing the suppressors has been very important. But what that also implies is that you have a batch of T cells out there that haven't truly been stimulated and could respond if you just take the brakes off. And I think that's going to be the next thing, is taking the brakes off. You block what you have to block, whether it's on the T cells or it's on the tumor, and then you allow the responses to occur. One must be careful with this, because if you take off too many of the brakes, you have a number of autoreactive T cells just hanging around that will attack your systems, and these have been the major toxicities we've seen with these particular therapies. So control the tumor by releasing your immune system, and no cats were harmed in the making of this particular production. How does this work at a antibody level? Because those are the actual, we call them drugs, are usually antibodies. What we have, as I kind of mentioned, the antigen-presenting cell stimulates the T cell with the MHC complex, as well as these co-stimulatory molecules. The T cell in response gets wound up, but also sticks out a molecule to say, okay, in case I need a kill switch, here it is. In case I lose control, here's that. So this could be, that leads to the inhibitory aspect of it, where the antigen-presenting cell has the PD-1 ligand, whereas the T cell has the PD-1 receptor. That will turn it off and deactivate it. The strategy is to use antibodies against one or both of those, and therefore keep that response on and keep it from being turned off. Tumors do this as well. Tumors have PD-1 ligand. They present, and that's what causes the T cell to attack. But if they have PD-1 ligand, they can also stimulate the PD-1 receptor on the T cells and effectively inhibit that. So once again, using the antibodies to block those on both sides of the coin seems to be a very popular therapeutic approach right now. And you're going to see a lot of that in the future. It is a multi-billion-dollar industry, so get in on that if you can. All right, to wrap things up, because I think I've gone a long time, I just want to point out that some collaborators are better than others. Jose Cuervo, Disaster Cat, and Kitchenwich are not necessarily good ones, but Tom Anschutz is. Also, the people who are truly responsible for this, Justin Hellwinkle, who's now a med student, he was instrumental in doing a lot of the work here. Solomon Ushi is now a resident at Mayo. He was the one behind the astrocyte story. Jamie, Mary, Dishley, and Tessa have been working with us for a long time. They're now, some of them are in med school. Yasmina started most of the proteomic work that I didn't talk about. My two bosses, Kevin and Aviva, and I want to thank Jen Maitland for keeping us out of IRB jail. And I want to thank the money, because that's obviously very important. So I want to thank them and thank all of you. And as it turns out, I'm not actually, if you have questions, you should be able to email me. What I would suggest you do is, I don't often respond to emails because I forget very quickly, so keep emailing me. Don't be afraid to keep emailing me. I'll eventually get back to you. So I want to thank you for that, and thank you for for letting me be a part of this.
Thank you, Dr. Graner, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do brain tumor exosomes end up in the bloodstream?
Yes, they do. I didn't mention this very much about the biomarker field that is exploding around these things. So the term liquid biopsy is often used, the idea being that if we can pull, say, blood out of somebody and then analyze components of the blood and say, oh, you have a tumor, or your tumor has these mutations, or you have this disease or that disease. We think of these things as soluble molecules, but it turns out that probably a lot of that is actually floating in these vesicle forms. So people are using proteins, but they're especially turning to the RNAs because those can be amplified. So the question from a brain tumor perspective is, can they get across the blood-brain barrier? And it appears that they can, and in terms of both ways, not just exiting the brain, but getting into the brain. So yes, they do get out. We identified them years ago. Johann Skog and Xander Breakfield's group did this years ago as well. So we know that they're out there. Part of the issue with the brain initially, the brain tumor initially, is that it kind of leaves holes in there, so things can escape the blood-brain barrier. But when you remove the tumor, that tends to heal, and yet these vesicles still manage to get out. So those are evident, those are present, and we can use those, we hope we can use those, as biomarkers of therapeutics in this case. Do we see changes in the microvesicle type and content and numbers, and is it responding to therapies that we're using to treat the patients?
Thank you for that answer. It looks like we have time for one more question. Our next question is, how can exosomes be used as vaccines at a practical level?
That's an interesting question. Generally speaking, these come from cell line sources, at least most of the academic studies. And that means you've got the cell lines available. And that's not going to necessarily be the case from a personalized perspective, say from a patient. The areas where we've managed to get them in large quantities would be from cancers that generate like ascites fluid, where you're able to obtain large volumes of liquid from a patient's stomach, basically. And those are also chock full of exosomes. Those have been used in clinical trials. That's from the tumor side of things. People have also used exosomes from dendritic cells as vaccines. So the idea is you take a patient's dendritic cells out of their blood, you grow them in culture, you throw antigen on them one way or another, and then you collect the exosomes that come off. We still do inject the dendritic cells back in, but there's all sorts of viability issues, freezing issues, activity issues. So the idea being that you can harvest the vesicles from the dendritic cells themselves and use those as vaccines. Those are sort of the two ways we think of that. As we start developing better bioreactor type growth phenomenon for these, and as we start to realize that maybe you don't have to have a personalized vaccine, we could have more general antigen sort of loaded into that, we may be able to actually engineer these things. And that's sort of the state of the art right now.
I would like to once again thank Dr. Michael Graner for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.